Hey everyone. I'm going to give you a little review. Chapter 1 in the Leninger textbook is basically kind of a review of sort of all the relevant biology that you would have had in other courses prior to this one. A little bit of biology and a little bit of, I think, a little bit of a chemistry review as well. So I'm going to go ahead and, since I'm not going to talk about it in class, I'm going to create a video where you guys can listen to and see if you have to review anything or just kind of maybe refresh your memory. But go ahead and sit back and kind of listen to the class. And there won't be any questions on exams over this material, but understanding a little bit of this material will probably help. So life arose on Earth about 4 billion years ago. It started off as simple microorganisms, and it required extracting energy from chemical compounds that were around, as well as sunlight. They use this energy to make biomolecules from the simple elements and compounds around the Earth's surface. There's a lot of discussion about that, and I think you probably have talked about that in biology classes in the past. What is biochemistry? Biochemistry describes the structures, mechanisms, and chemical processes shared by all organisms. So biochemistry is one of the good things about biochemistry is generally all the biochemistry is the same no matter what the organism is. There might be some modifications for how that organism lives and survives, but they're just they're kind of tweaks or modifications of the standard process. So let's go ahead and first talk about sort of the cellular foundations. We know that the whole principles of biology is that the main unit of biology is cells, and you have multicellular organisms, like we're made of probably billions or trillions of cells, and it's all based on this whole cell theory. And we're going to go a little bit of that. Bacterial cell, if you look at it, has very little organization. It has a nucleoid in the middle that has all the nucleic acid material. You have these ribosomes floating around. You have a cytoplasm in the middle. You generally don't have any membrane structures at all within the microorganisms. While if you start to look at eukaryotic cells, we start to see a lot of substructures. We see cytoplasm, we see a nucleus that has the genetic material in it that's surrounded by two layers of membranes. You have mitochondria, which are double layers of membranes as well. And you have this whole endomembrane system, which we'll go into more detail about how it forms and what's involved in it in a future lecture. But you see there's the cell, and the animal cell is significantly larger. Look that, a bacterial cell is maybe a micron long, while a eukaryotic cell is maybe 20 to 50 microns long. So they're significantly larger. So the plasma membrane, it's sort of like, I like to think of the plasma membrane as that sort of that sac that holds all the cellular contents together. So it's composed of a lipid and protein molecules. We'll see this fluid mosaic model we'll talk about in a future chapter. It's a thin, flexible hydrophobic barrier around the cell. It's embedded with transport proteins, receptor proteins, membrane enzymes, all sorts of things to help that cell interact with the environment and get information from the environment. So think about the plasma membrane as sort of this sac that holds everything, all the contents together. Now the cytoplasm is that volume that's held inside the plasma membrane. It's composed of cytosol, which is an aqueous liquid, and it has a bunch of suspended particles, some of them as large as mitochondria, ribosomes, proteasomes, but it has these smaller substructures as well. And cytosol is the concentrated solution that contains the enzymes, RNA, amino acids, and nucleotides. There's just a little bit of difference there. Cytoplasm is the whole thing, and the cytosol is just that concentrated liquid. The genome is this complete set of genes that are composed of DNA, and DNA has all that molecule in it. And formerly grouped in prokaryotes, it's this nucleoid, and in eukaryotic cells, it's membrane bound. All this nucleic acid, the genome is present in this enclosed nucleus. Cellular dimensions are limited by diffusion. Whenever you get down to these small sizes, the rate at which things diffuse wind up being the rate limiting step for things to happen. So cells generally have certain limits to how large they can get. And in this picture, you see, in this slide, you see there's a picture of some human lymphocytes. And you see they all have some different structure on that. But cells are microscopic. Plant and animal cells are 5 to 100 microns in diameter. Unicellular microorganisms we talked about before is 1 to 2 micron long. The limit of cell size is likely set by the rate of oxygen transport in the cell. As the size increases, the surface to volume ratio decreases, and that makes the diffusion rate even more defending. That kind of at that certain point, the diffusion rate isn't sufficient enough to get oxygen to all parts of the cell. So at that point, the cell really can't get much larger than that. You'll probably cover a lot of that sort of cell size information if you take physiology classes in the future. Cellular physiology may talk about that as well. Now, whenever I was in your shoes, we talked about sort of everything was sort of plants and animal kingdom, and it was this big debate as to where do the microbes fit and things like that. So now there are 3 domains of life that have been kind of discussed in great detail. There's the bacteria, which can, it's a number of different things, microbiology covers it. There's these archaea, which are these sort of microorganisms that generally live under extreme conditions, like high salt, high temperature, high amount of methane, or the lack of oxygen, things like that. And then the eukarya, it includes all the animals, fungi, plants, ciliates, a whole bunch of these things. So there's a whole bunch of these other things here that are also eukaryotic cells. So we have these 3 domains of life. Organisms belong to these 3 distinct domains. So bacteria inhabit the soils, surface waters, tissues of living and decaying organisms. We have bacteria living inside of us, for example. And if they weren't there, we would have difficulties. Archaea live in extreme environments, and eukarya are basically all eukaryotic organisms. Generally, they're multicellular, but there are a few of them that are not. Archaea and bacteria are subgroups are distinguished by their habitats. Archaea live in extreme situations, and bacteria live under less extreme conditions. But some of them will be aerobic, meaning they need a plentiful supply of oxygen. So any aerobic organism needs to have lots of oxygen present. And it uses fuel it takes electrons from O2 to get energy. Anaerobic organisms devoid of O2, the organisms transfer electrons from nitrate, sulfate, carbon dioxide. Within this group, there are obligate anaerobes that die when they're exposed to oxygen, and there are facultative anaerobes which can live with or without oxygen. They can live under either of those conditions. So those are the 3 sort of groups of organisms we have. Either they require oxygen, or they die in oxygen, or they can live without oxygen. So organisms differ greatly in their sources of energy and biosynthetic precursors and things like that. So these photoauxotrophs, they trap and use sunlight. So photo meaning light. Okay, chemotrophs are used to derive energy from oxidation of chemical fuel like sulfates, nitrates, things like that. And like there's a lot of organisms like that. A lot of the archaea are chemotrophs. Autotrophs, they can synthesize all their biomolecules directly from carbon dioxide. There's a lot of plants and cyanobacteria and things like that that are auxotrophs. And there are heterotrophs that require some preformed organic nutrients made by other organisms. We're a heterotroph. We eat food to get energy. And so we're a heterotroph. So classifying organisms by their source of energy. This is another way to kind of look at how these organisms come about. So you can sort of, you have this group of all organisms here in this area here. Depending on the energy source, if it comes from light, you have phototrophs. And the carbon source is carbon dioxide. You have photoauxotrophs. They use water. You can use water to reduce CO2. There's two different mechanisms. We won't probably go into them this year, but oxygenic photosynthesis, plants, algae, cyanobacteria do these. And anoxygenic photosynthetic bacteria, green and purple bacteria and things like that, do not use water to help reduce carbon dioxide. And here, carbon sources, other organic compounds, they're photoheterotrophs, meaning those organic compounds have to come from somewhere else. So they're a heterotroph, but they also use light, so they'd be referred to as a photoheterotroph. And they're green non-sulfur bacteria and purple non-sulfur bacteria that fall into this category. So these are all that use light. Now the ones that use chemicals fall in the other categories. It's the carbon source is CO2. They're chemoautotrophs. They basically make their own hydrogen, sulfur, nitrogen, carbon dioxide oxidizing bacteria, can do all those things on their own. Some that require the organic compounds. We'll talk a lot about those as well as courses about these organic compounds. So chemohomotrophs, there's the final electron transfer, the final electron acceptor. If it's oxygen, all animals, most fungi, protists, bacteria, organisms like ourselves. If the final electron acceptor is not oxygen, it's an organic compound. It's sort of a bacteria such as Lactococcus lactis and things like that. And you have some that use inorganic compounds like some of the Pseudomonas denitrificans. So you can see that there's a variety of ways that organisms kind of get their energy and metabolize their energy. And it's kind of broken down into, is it light or chemistry, and then whether or not how oxygen and water is being used in that metabolism. And those are the basic differences between all the organisms. When we start talking about the biochemistry now, a lot of those other things will wind up being very, very similar. Let's talk about this. Let's talk about the sort of structure. We referred to these bacteria and archaea cells quite a number of times. Let's look at those structures a little more closely. This will be a physics figure in your textbook. So the cell envelope, which is this green thing here, basically holds the contents of that organism together. And within that organism, you'll see the nucleoid, which is all this yellow material, the DNA in there. And later on, we'll talk about how long that DNA is and how small the cell is. And you might be kind of amazed how much DNA there is there. They potentially have ribosomes in here that'll make protein, because you have the DNA will be used to make messenger RNA, which in turn makes protein, and ribosomes are required to do that, and they actually have both protein and RNA present in them, and we'll go into that later on in the semester as well. But to be able to move around, a lot of bacterial cells have flagella that propel it through their surroundings, and some of them have these pili that allow them to connect and adhere to surface of other cells. And some of the pili are involved in some of that transfer of DNA in things like multi-drug resistance and things like that are kind of transferred from organism to organism by way of these pili proteins and these pili. Now we start looking at, we start to see some structural differences between some of these organisms. If you've had microbiology, you've already heard about gram-positive and gram-negative bacteria. It's basically a way that these organisms wind up taking certain stains. But gram-positive organisms have a polysaccharide on the surface. It's a solid layer on top. There's some glycoproteins on the surface, peptidoglycans, and these lipoproteins attached to them in the membrane. So it's got kind of like a coating on top of the membrane. While gram-negative bacteria have an outer membrane that have porins, have lipopolysaccharide on the exterior. They have a periplasm that has a number of different structures in it, has peptidoglycans, things like that in it, lipoprotein, and there's a cell membrane that has all these sort of transporters and things like that. Now, some of the archaea are sort of like gram-positive bacteria, except they don't generally have the sugar coatings on the outside. their peptidoglycan. It's not really the same. It's a little bit different structure, and we might talk about that a little bit later. So they all sort of have some similarities, but they all have their differences. Now, E. coli cytoplasm contains ribosomes, enzymes, metabolites, cofactors, all these sorts of things. And the nucleoid, which is that the massive DNA in the middle, is one big circular DNA molecule. Almost all bacteria have this. But it was found in the early days of sort of molecular biology that there could also be these plasmids, which are smaller circular segments of DNA that confer resistance to toxins and antibiotics in the environment. And we'll talk a little about how we've taken advantage of and used plasmids. But plasmids are naturally within these organisms, and they have a little bit extra coding regions for the organism to take advantage of. Eukaryotic cells can have a variety of membranous structures, which can be isolated for structure. For study, there's some mechanisms we'll probably talk a little bit about how we isolate and we can fractionate various different parts of the cell. But we have mitochondria, which is that energy extracting part of the cell. There's basically you can make all the energy containing molecules can be made in the mitochondria. There's endoplasmic reticulum and Golgi, which are these membrane structures in the cell which play central roles in the synthesis and processing of lipids and membrane proteins and exported proteins. Peroxisomes are the site of oxidation of long-chain fatty acids, the detoxification of oxygen-reactive species. Some people might remember, you might have seen the acronym ROS, and they're kind of regulated in the peroxisome. Lysosomes have digestive enzymes where it breaks down the complex carbohydrates. Peptidoglycans, proteins are all degraded into their inner components so they can be recycled within the cell. And then within the cell, in the cytosol, there are these granules or droplets containing nutrients like starch and fat. Plant cell organelles have the same thing as eukaryotic animals have. They have this large central vacuole that contains large quantities of organic acids and things like that. And these organelles are really there for structural strength in the plant as well. And chloroplasts, which everybody knows is the green part that harvests light to make sugar from carbon dioxide. So let's look at what the cells look like. Here on the top portion here we have an animal cell. On the bottom portion here we have a plant cell. And you'll see there's a lot of very common things. You start looking, you see these membrane structures look very similar. You see mitochondria in both of them. You have a cytoskeletal structure, which we'll talk a little about later. Lysosomes, which break down things. So you see they look very similar, but plant cells have this big central vacuole, and they have chloroplasts or plastids, which are involved in sort of fixing CO2 and things like that. Just to let you know, there are some techniques out there that people have used over the years, and they have found that you can sort of isolate various different subfractions of a cell. Biochemists like to go ahead and analyze what's going on within a cell instead of just looking at the whole organism level. They like to break it down into very simple little parts. And to do that, we frequently need to break down the cells and find out which component does what activity is where. And there's a technique called differential centrifugation, which is commonly used. And you can isolate and separate organelles by their different size and sedimentation rates and all this stuff. This is something you don't really ever have to memorize, but just know that, low speed centrifugation, you get rid of some of the big things. You start to go greater and greater forces, and you start to get smaller little organelles and pellets. And that final pellet has ribosomes and large macromolecules. You have microsomes and a fraction, which a lot of people use for studying like cholesterol metabolism and membranes, things like that. So we can have there are mechanisms in which you can use and sort of isolate a particular part of the cell that you want to use. And centrifugation can do that for you. Cytoplasm is organized by the cytoskeleton, and it's highly dynamic. It's always inside the cell is always changing, and there's a lot of activity going on. And there's a complex series of proteins inside the cell called the cytoskeleton, which is this 3D network of protein filaments within the cells. They're composed of actin filaments, microtubules, intermediate filaments. All these things have slightly different sizes, slightly different chemistries, and slightly different functions, which we'll get into a little bit later. If you take sort of cellular biology, cell biology, or cell physiology, I'm sure you'll get into those in a little even more detail. Filaments undergo constant assembly and disassembly in the cell, so they're being made and taken apart, so that leaves sorts of dynamic aspects. And they lead to some very kind of interesting pictures of cells as well, like we can see here. There's a technique used where you can put a fluorescence tag onto a protein, onto an antibody, which will bind to certain proteins within certain structures. So we here have, like in this plate A, all the actin filaments are here in red, microtubules are all in green, and the chromosomes are all kind of in this blue color. So you can see how the cell is organized. And you see that, actin and microtubules sort of create the sort of the structure of the cell. Another more elaborate experiment has been done where they have 5 different things being isolated. This is probably as the cell is starting to divide. You have microtubules in green, kinetophores in this kind of an olive color. You can see that in a couple of places. Chromosomes are in blue. You see them all lining up here. Centrosomes in purple, which you don't really see. You see here and here. And then intermediate filaments surrounding the cell, getting ready for that cell to divide. So you can actually kind of see how the cell is dividing and what things are in what areas and that sort of thing. So, it's very nice that we've had a lot of these little tools to dissect out the little portions of the cell, and then we got them all by breaking them down and isolating them and making tools to detect them. Now, I made reference to the endomembrane system. We'll go into it in much more detail in chapter 11, I think, in our book. But it basically segregates the cell into different sections. So there's different metabolic processes that happen in certain places, and the endomembrane system kind of helps that. And endomembrane system is also involved in exocytosis, which is exocytosis, meaning outside the cell, and endocytosis, meaning inside the cell. So exocytosis is the export of things outside the cell. Endocytosis is sucking things up from the media and going into the cell. They involve in membrane fusion and fission. Think about it. You might have 2 cells fused together. In fission, you see when cells are dividing, they divide into 2 cells. And these provide the paths for the cytoplasm and surrounding media and all that. And we'll talk a little about how that's partially regulated and how that all works, albeit towards the end of the semester. So we've talked about different kinds of structures, and part of what I've heard it being used a lot in different biology courses, kind of use some of the same terms, but sometimes they don't. And I want to make sure you get that these are the terms that I'll be using during the semester. So supramolecular structures, things like the plasma membrane, chromatin in the chromosome, the cell walls that separate them. Those are all supramolecular structures, so they're large molecules making these very big complexes of lots of large molecules. Then we have these micromolecules. You have proteins, DNA, cellulose, and things like that are involved in these processes. And to be able to make these, we have these monomeric subunits that are involved in making them. And in each section be going into during the semester, we'll talk about the subunits first, And then we'll talk about the macromolecules they do, and then we'll talk about some of these supramolecular structures. So we're going to do the classic biochemical approach, look at the monomers, look at the macromolecules, and then the supramolecular structures. So this kind of gives you the organization of how we're going to be doing the class, and we're going to be going from right to left instead of left to right. Terms that will get used a lot. In vitro is Latin for in glass. And in glass means in a test tube. So separate it out, and classic biochemical things, we use individual, we break things down to simple complexes, simplest possible complexes, and then build upwards. In vivo means in the living, so that's within living cells, within the organism. Molecules may behave differently in vivo than in vitro, but in vitro studies frequently allow us to predict and be able to lower the possibilities are to understand the in vivo kind of things. We can start seeing here inside the cell, you see all these different aspects of the cell again. We have these kind of nice illustrations. That was the sort of cellular things. Now let's go ahead and talk about sort of the chemical foundations that we need to know. And this is the part that a lot of you may not remember very much, and this is one of the reasons you'll find it one of the reasons why we kind of expect you to have organic chemistry before coming into this class. Let's go ahead and go sort of broadly in chemistry. All these sort of salmon-colored elements are all things that are bulk elements within the cell and within macromolecules and supramolecular structures. And then we have these other trace materials that are here in dark yellow, which are involved in other processes. A lot of these things that are in yellow will help the cell do things. So they're kind of we refer to them as being cofactors. So a lot of enzymes and things like that need like magnesium or iron or something like that to be able to do their work. Biomolecules are compounds of carbon with a variety of functional groups. You probably learned a lot of these things in organic chemistry. We'll breeze through these pretty quickly. You have carbon-hydrogen bonds or single bonds. You have carbon-oxygen forming single bonds. You can actually have oxygen forming double bonds to carbon. Happens quite regularly. You can have carbon-nitrogen bonds. So you have carbon-nitrogen bonds like this, forming like amino groups and imide groups and things like that. You have double bonds between nitrogen and carbon. Carbon can form single bonds between other carbon atoms, double bonds or triple bonds. All these things can occur in biological systems. Triple bonds are not as common. Double bonds are fairly common, and single bonds are all over the place. A lot of this geometry of the molecules are very, very important. In organic chemistry, you may have been told about a lot of these orientations. Like you have carbon, it can make 4 bonds, and each of these bonds are equal distance from each other. So they're all 109.5 degrees apart. If you go ahead and, if you're clever mathematically, you can come up with a triangle and figure out what all the bond angles would be. But it's 109.5. You can have these bonds are freely mobile. So you can have, if it's a carbon-carbon bond, for example, it can rotate around each other. It's free rotations around that single bond. But if you have a double bond, you wind up everything winds up sort of, the 2 carbons and everything they're bound to all wind up being in the same plane. So they wind up being planar. And that really constrains some of the structures of some of these molecules. And we're going to see later on how those kind of bonds kind of make everything, help constrain some of the structures that will actually allow us to predict some 3D structures and things like that. So common and functional groups, be aware we're going to probably see these pop up from time to time. You might want to be kind of familiar with them, methyl ethyl groups, phenyl groups, Guanidinium groups were arginine, for example, imidazole, histidine, sulfhydryl groups, cysteine, disulfides, two cysteines connected to each other, anhydrides, two carboxylic acids attached. We'll find out those are very high energy bonds, like for having phosphates attached to each other, acetyl groups, things like that. Carbonyl, carboxyl groups, amido groups. So we can see all these things all associated with each other. So we have these things that are thioesters because sulfur has a lot of sort of the same chemistry as oxygen, but some little differences here and there. Phosphoanhydrides, mixed anhydrides, things like that can all occur. But one thing about it is we talked about all those groups. I went through them really fast because you probably have been introduced to all of them before. But what's really kind of interesting is a lot of biomolecules have many of these functional groups. So they're polyfunctional. Like look at, this is acetyl coenzyme A. It's a cofactor that's used in a lot of different aspects of biology. You see here there's thioesters, there's amido groups, there's hydroxyl groups, there's phosphoanhydride group, there's an imidazole-like group, there's amino groups, and phosphoryl groups, all working together to make a molecule that have a certain specificity and a certain structure. So cells contain a universal set of small molecules. So some of the central metabolites are common amino acids. You might know those, the protein amino acids or the essential amino acids, things like that, nucleotides, sugars, and phosphorylated sugars. And then there's a whole bunch of monodiene and tricarboxylic acids that are involved in doing things. Those are all common to almost all organisms, particularly all amino acids and nucleotides are all in common. And sugars, phosphorylated derivatives are pretty common as well. Secondary metabolites wind up being specific to the organism. Like you may be drinking a cup of coffee right now, and that's extraction of coffee beans of secondary metabolites that are very specific to coffee beans, for example. They're very specific to an organism. And you're going to see throughout this semester, I want to talk about sort of disciplines and studies and names. Humans love to put names on things. We're talking about here, like, for example, the metabolome is the entire collection of all the small molecules in a given cell under certain conditions. And the study of metabolomes is called metabolomics. And it's a characterization of the metabolome under very specific conditions. And we've gotten a lot of tools, a lot of computers have really allowed us to be able to understand a lot of things that are happening metabolically. In the past, we studied individual pathways. Now we can actually study many, many different pathways. We'll go into some of those. A lot of pathways and metabolism will be covered in biochemistry too. So we won't be talking about a lot of metabolism. We've been talking about quite a bit in this class. Macromolecules are polymers with molecular weights above 5,000 that are assembled from relatively simple precursors. Proteins, nucleic acids, polysaccharides all fall in this category. We'll use the term oligomers to a certain degree. I have a tendency not to use them very much because it's kind of a vague term, but shorter polymers are called oligomers. Informational macromolecules, a name for proteins, nucleic acids, some oligosaccharides, because they have information-rich subunit sequences. Some of these molecules are made, it transfers information, like what the cell does, what the cell needs to do, what it needs to react to, and all these things come from these informational macromolecules that circulate around. Proteins. We're going to have a whole chapter about protein. Well, not a chapter. We have about four chapters. We'll talk about different aspects of proteins. Proteins are long polymers of amino acids. They can function as enzymes, structural elements, signal receptors, transporters. They have a huge number of functions. The sum of all the proteins functioning in a cell is referred to as the proteome. And the study of proteomes is called proteomics. A lot of proteomics is oriented to defining which proteins are present in cells at certain points. Those of you taking microbiology here, whenever you've done that final ID of your bacteria, you've used a proteomics approach. The MALDI mass spec approach is looking at the proteome of the cell to identify that cell. Nucleic acids, another large macromolecule, is DNA and RNA polymers. They're made, with nucleotides connected together in a particular way. Some RNA molecules have structural and some even have catalytic roles in supramolecular structures like in ribosomes and things like that. The entire sequence of a cell's DNA and RNA is called the genome, and the study of genomes is called genomics. Polysaccharide molecules. The polymers are simple sugars. They can be energy-rich stores. They can be structural components of cell walls and plants and bacteria. Extracellular recognition helps cells bind to other cells, all dominated by polysaccharides. And the glycome is the complement of all the carbohydrate-containing molecules. And the study of those, which I don't have on the slide, is called glycomics. Lipid molecules. Lipids are the last sort of group of molecules we'll talk about. Lipids have water-insoluble hydrocarbon derivatives. They'll make up structural components of membranes. They're energy-rich fuels. A lot of pigments, like the red colors of cardinals, yellow colors of other birds, all come from some of these lipid-like molecules. And a lot of intracellular signals come from lipids. All the lipids in the cell is called lipidome, and lipomics is the study of all those. So let's sort of talk about let's look at some of these molecules and look at some of these structures. I've talked about them in general. Let's look at some of these molecules. Like here, amino acids all have a very similar structure. You'll have this referred to as alpha amino acids. So you have a carboxyl group, and you have this carbon that's alpha to that carbon has a nitrogen group, right? An amino group. So these are all amino acids. And then you'll have, and they all have an alpha hydrogen attached, and they'll have an R group attached. There's only one molecule that's exception from this, but this is what amino acids generally look like. Now, nucleic acids will have these nitrogenous bases. You see there's a lot of nitrogen in them. They're attached to a sugar, and they're attached to a phosphate group. So you see these molecules happen in a number of different places. We'll talk about them in great detail. Sugars, I'll have this structure sort of like this, and lipids wind up looking like, maybe you'll have nightmares going back to alkanes and organic chemistry. They'll have these fatty acids. They have choline in different head groups. They have glycerol attached to them as well. Just to let you know, sort of these major classes of biomolecules. This is an E. coli. This is a table that you would never want anybody to ever really memorize, but this is sort of the number of species and the percentage of the large groups. For example, water is about 70% of the cell. And there's only one kind of water molecule. Proteins is about 15% of the cell, and there's about 3,000 of them present at any one time in the cell. Nucleic acids, DNA, there's one. It can be 1 to 4% of the cell. RNAs, they're about 6% of the cell, and there's probably about 3,000 of them. There's all the mRNAs that make the proteins. There's RNAs for the structural, they're making the ribosomes to transfer RNAs. So there's more RNA diversity than there would be of proteins because they're involved in making the proteins. Polysaccharides are about 3% of the cell. There's probably about 20 different groups of them in the cell. Lipids are probably about 50, but you'll see later on that number may actually be really much smaller than it really might really be. Monomeric subunits, intermediates, there's probably 2% of those, and there's probably about 2,600 of those, and there's probably about 20 different inorganic ions. So there's a lot of different things in the cell, and you see a lot of these things we'll spend a lot of time talking about are very small percentages of the cell with water dominating the cell. So three-dimensional, when we wind up, you may have sort of gotten an idea that it's the structure of these molecules that really determine a lot of function. And we'll talk about, and you probably remember chirality or stereoisomers and stereospecificity from organic chemistry, but the configuration is the fixed arrangement of atoms in a molecule. Stereoisomers are they right-handed, left-handed molecules. They have the same bonds, the same formula, but they're different hands, right? Stereospecific in biology and biochemistry, a lot of these things, one isomer will be reactive while the other will not be, for example, because the stereochemistry in living things is very, very specific. You'll see like all the amino acids are all L amino acids. Almost all the sugars are all D amino acids. I mean, D sugars, that sort of thing. But here's when we look at some stereochemistry. Here's, alanine. You see there's the carboxyl group. There's the amino group. There's that chiral carbon. It looks like it has 1, 2, 3, 4. It has 4 different groups on it. You have the ball and stick form here in your space-filling molecule. You can see it here. And you'll find that sometimes all these different ways to draw structures all have their reasons. And you'll find that sometimes one is easier to understand than the others, depending on the case. So chiral centers, probably learned about this in organic chemistry. You look at them, your right hand and left hand are mirror images of each other. So you put your hand up to a mirror. What you see in the mirror of your right hand, for example, will look like just like your left hand, and your left hand will look like your right hand. So what we can do is realize that in biological molecules, that can be pretty complicated. They can have many different chiral centers, which the more chiral centers you have, the more possible stereoisomers you have, where if a molecule has 2 to the n, so stereoisomers, n is the number of chiral carbons. So if there's 2, and it's 2, so there's 4 isomers. If it's 3, it'll be 8 isomers, things like that. But keep in mind that the original molecule looks like this in the mirror images you see. And keep in mind that we're going to be talking about those quite regularly through the semester. Enantiomers and diastereomers. Enantiomers are mirror images of each other. So they're mirror images of each other. So you have this possibility here. You have these 2 sets of enantiomers, but the chemistry of all these compounds are basically the same. It all is about configuration around different carbon centers. So you have diastereomers or stereoisomers that are not mirror images of each other. So not all stereoisomers are mirror images of other molecules. Okay, keep that in mind. So as soon as you start getting to multiple, stereospecific atoms within a molecule, some of those generic rules kind of disappear. Enantiomers have nearly identical chemical reactivities, but different in optical activity. Racemic mixtures, equimolar versions of 2 enantiomers show no optical rotation. So if it's purely right-hand or left-handed molecule, there's ways where you can use optical, polarized light, and it'll bend light a particular direction. Racemic mixtures don't bend that light. So you have these different things, here you can look at it, you can name them, and there's some naming systems in there. And for biochemistry, you don't really need to know these, but this is sort of that connection to kind of remind you about organic chemistry. So conformation. So you can see these conformations of molecules. We talked about carbon-carbon bonds are freely rotating, but in reality, there's different potential energies in different configurations. If everything's all lined up, you have that sort of repulsion of protons to protons. They kind of want to get as far apart as they can. So you see that a higher energy conformation of the protons are lined up with each other, and whenever they're sort of in between each other, like here, are energy minima. And we'll have a look at kind of these kind of energy potential curves off and on during the whole semester. Configurations. So we have, and we're going to talk about a little bit more. You may not have talked about these in organic chemistry so much, but geometric isomer, cis-trans isomerization, things like that. Like you have malic acid that have the carboxyl groups on the same side as from the double bond where fumaric acid is across. So the same side we refer to as cis, and across would be trans. A lot of organic chemistry will use sort of the older organic chemistry nomenclatures. In some cases, the organic chemistry nomenclature is very specific, but it doesn't generally get used as much in biochemistry. You have these cis-trans, like in being able to see and absorbing light. You see there's cis-trans retinol with the presence of light forms all trans retinol. So you can see it's a configuration change. The molecule itself compositionally doesn't change, but structurally changes. And interactions between molecules are very, very stereospecific. Here, this is a 3D structure of a protein. This is the surface model where you can see all the atoms there. And you see in this pocket, you see something that, you see the stick drawing of a substrate. It fits in just perfectly. And so a lot of enzymes and things like that all have these pockets that are very, very stereospecific. That'd be a key thing. And just to go ahead and go along with that, so you see it, a lot of people use aspartame in colas and as a sweetener. And here's the structure of aspartame. It's a aspartyl phenylalanine methyl ester. This one configuration right here is very sweet, but the other isomer here, you see the other isomer here, where you see the phenylalanine is the other molecule. This molecule tastes very, very bitter. So you can see there's some extremes of how it might react. I think here's a compound. I think it's an anti-anxiety drug. I don't remember for sure. It's therapeutically very active. The isomer here is completely inactive. It doesn't have any biological activity at all in an organism. So it's just because of a stereocenter. And it's probably because it's not binding correctly to a receptor molecule or an enzyme.